In this lecture, we're going to discuss recombinant DNA technology. Following this lecture, you should be able to describe recombinant DNA, list the uses for recombinant DNA technology, identify the steps used for cloning and describe the theory of each step, describe the use of restriction endonucleases in recombinant DNA technology, list some examples of recombinant DNA technology, describe gene therapy, and discuss concerns regarding the use of recombinant DNA technology. So recombinant DNA technology is using recombinant DNA, and recombinant DNA is created by combining DNA sequences that wouldn't normally occur together. So this is a form of genetic modification. When you have recombinant DNA, you would make a recombinant protein, so that protein that's derived from that recombinant DNA. Recombinant DNA technology was made possible after the discovery of restriction endonucleases, and recombinant DNA technology is also referred to as genetic engineering as well as cloning. And we're going to discuss the theory behind recombinant DNA technology and cloning. There are various uses for recombinant DNA technology, such as manufacturing hormones and chemotherapeutic agents. There are numerous agents that are made using recombinant DNA, such as interferon, insulin, factor VIII, factor IX, human growth hormone, erythropoietin, angiostatin. Recombinant DNA technology is used for gene therapy, and this is a multi-billion-dollar industry. So molecular cloning or recombinant DNA. It's a genetic engineering technique that allows you to construct novel DNA molecules by joining two different DNA sequences. So joining DNA sequences from two different sources. So when you use recombinant DNA technology, you usually are going to use a vector, and there are various vectors that can be used. Plasmids are one of the more common vectors used, especially in research laboratories. So plasmids, you would insert different DNA into your plasmids, and the DNA that you're going to insert should be less than 10 kilobases. Also, some vectors are bacteriophages. Bacteriophages, you can insert approximately 25 kilobases of DNA. Bacteriophages are commonly used for gene therapy. Plasmids are you could put larger pieces of DNA into a plasmid, 35 to 45 kilobases. YACs or yeast artificial chromosomes, you can put 1 to 2 megabases. So those are for putting in huge pieces of DNA. So why do we use vectors? Well, vectors have things that are absolutely essential for recombinant DNA technology. For example, we're going to talk about the plasmid because that's one of the more common vectors used. Plasmids have an origin of replication, and that origin of replication in your plasmid is going to depend on what organism you put your vector into. So a lot of times we use E. coli for cloning. So the plasmid would have the origin of replication for E. coli. Also, you want to have a selectable marker, which is usually an antibiotic resistance gene. So you can identify that you actually have your vector that contains your recombinant DNA. If you didn't have that selectable marker, you wouldn't know if you actually have the correct vector. Also, there's a cloning site in what's called a multiple cloning site, and usually this is going to have several restriction endonuclease sites. So it depends on what vector you use. Is the different vectors are going to have different endonuclease sites. So for example, this vector right here has a Hindi3 site, a PST1 site, an EcoRI site, a SAL1, a BAMH1, and a KPN1. So if you absolutely needed to cut your DNA of interest with an enzyme that isn't located in this particular plasmid, you would want to choose another plasmid. So you have to choose the right plasmid or make a plasmid for your specific needs. So remember, we talked about plasmids before. Plasmids are small circular double-stranded DNA that usually carry an antibiotic resistance gene, which is important for recombinant DNA technology because you need that marker. Plasmids are very common in bacterial cells. So bacterial cells commonly have numerous plasmids in them. These plasmids can readily pass from one bacterial cell to another bacteria, even among different species. Plasmids are autonomously replicating and they are extrachromosomal. Now remember, your restriction endonucleases are enzymes that cut when they recognize a specific 4 to 6 base pair restriction site. So each restriction enzyme is going to have a different restriction site, and each enzyme is going to cut at a specific location within this sequence. So for example, this slide shows EcoRI, and EcoRI is an enzyme that's going to recognize the sequence GAATTC. And whenever it sees those 6 nucleotides in that exact order, it's going to cut between the G and the A. So what you would do in recombinant DNA technology is you would cut your vector as well as your gene that you're trying to clone with the same enzyme. So for example, you would cut your plasmid with EcoRI, and you would cut your gene of interest with EcoRI. That's going to cut the GAATTC sequence between the G and the A, and it's going to generate sticky ends. So then what you can do is you could take your plasmid that's been cut and you take your gene that's now been cut out of your genomic DNA, and you could combine them together. So here it is again, just showing your plasmid that's been cut, and it has now sticky ends, and your gene of interest, double-stranded, that's now been cut and has the homologous sticky ends that can then be put together. And here we are again. We have 2 different molecules, molecule A, molecule B. In this case, we're digesting with the enzyme BAMH1. BAMH1 recognizes the sequence GGATTC, and it's going to cut between the 2 G's. So it's going to generate sticky ends on both molecule A and molecule B, and how you would then put these 2 together is with the enzyme ligase. So remember, we talked about ligase before. Ligase acts like a glue, and it's going to seal together nicks and things in DNA. Well, for recombinant DNA technology, ligase is going to attach your sticky ends together that have the corresponding nucleotide sequence. When you take your 2 molecules, cut them with an enzyme, and put them together with ligase, you end up with recombinant DNA because you have DNA from a completely different source attached to other DNA. So here we have our plasmid, you're going to cut with EcoRI in this example. So you're going to cut your plasmid, and it's going to cut between the G and the A. You're going to cut your DNA of interest between the G and the A, and then you're going to ligate your gene of interest into your plasmid. And here's what you would end up with, and this would be considered recombinant DNA because you have plasmid DNA mixed in or attached to DNA from a gene from a completely different source. So again, lots of pictures in this lecture because this is very visual. So you have your plasmid, you have your marker, which is going to usually be an antibiotic resistance gene. You're going to cut your plasmid, usually generating sticky ends, but remember we do have restriction endonucleases that cut, making blunt ends. You would also cut your gene of interest. You're then going to ligate in your gene of interest into your plasmid, making recombinant plasmid. The next step is actually taking that plasmid and transforming it into bacteria. So remember, your plasmid has an antibiotic resistance gene, and now it's a recombinant plasmid because it has DNA from another source ligated into it. You're then going to transform that plasmid into a bacterial cell. Then what you're going to do is take that bacteria and make a lawn of growth on an agar plate that contains the antibiotic that matches the plasmid's antibiotic resistance gene. So any bacteria that can grow up on that agar with antibiotic contains the plasmid that has your antibiotic resistance gene, which would indicate that that bacteria has your recombinant plasmid. So this whole process is referred to as cloning because you're making clones of that recombinant DNA using bacteria. When you put your recombinant plasmid into your bacteria, such as E. coli, E. coli doubles every 20 minutes, 20 to 30 minutes. So you are going to make many clones of your recombinant plasmid in that bacteria. So once again, you take your bacteria that contains your plasmids, you streak it out on agar that contains the appropriate antibiotic, and if that bacteria is able to grow, it means it contains the plasmid with the antibiotic resistance gene. So here's a summary again. You have your plasmid with your cloning site. You cut with the appropriate restriction endonuclease. You also cut your DNA of interest with the same restriction endonuclease. You're going to ligate the gene of interest or foreign DNA into your plasmid DNA. You're then going to transform your plasmid into bacteria, and this is done using either heat or an electrical current to force that plasmid into bacteria. You're then going to grow your bacteria in the presence of antibiotic, and then you can isolate your plasmid from your bacteria. So here's an example of how you would make a chimeric plasmid. So on the top left, you have a plasmid that contains the ampicillin resistance gene. On the right, you have a different plasmid that contains the kanamycin resistance gene. So let's say you wanted to make a plasmid that has both the ampicillin and the kanamycin resistance gene. How you would do this is you would take your plasmid with your ampicillin resistance gene. Now if you look at this plasmid, the entire plasmid is 4,539 base pairs in length. There's a BAMH1 site and a HND3 site on this plasmid. If you cut the ampicillin resistance the plasmid with the ampicillin resistance gene with the BAMH1 and HND3 enzymes, you will remove a 784 base pair fragment from that plasmid. If you cut your plasmid that has kanamycin resistance gene, and that plasmid is a total length of 4,207 base pairs. If you cut that also with BAMH1 and HND3, you are going to remove a 1,875 base pair piece of DNA containing the kanamycin resistance gene. Now, you would have to use both the BAMH1 and the HND3 site because you need to remove that entire kanamycin resistance gene and put it into your other plasmid. So that is why those two restriction enzymes were used in this example. So there's a lot of careful designing that has to be done in recombinant DNA technology. So okay, you've cut out a 784 base pair piece from one plasmid. You've cut out a 1,875 base pair piece from another plasmid that contains the kanamycin resistance gene. And now you can ligate in that kanamycin resistance gene into your plasmid because now your plasmid has the nice sticky end from BAMH1 and a sticky end from HND3, and so does that kanamycin resistance gene from your other plasmid. So what you make is a chimeric plasmid that contains both the ampicillin resistance gene and the kanamycin resistance gene. And your chimeric plasmid now is 5,630 base pairs. So you should be able to look at a plasmid and be able to determine the size of the plasmid depending on what's been removed from the plasmid and what size gene has been put into the plasmid. You can then isolate your plasmid from your DNA, and there are kits called plasmid extraction kits. You just grow up your recombinant E. coli that contains your plasmid, and you put it into a spin column and you spin and you add some reagents to it, and within about 10 minutes you have nice clean plasmid DNA. You would then cut your plasmid that you've just isolated from your various clones from your E. coli plates, and if you cut it with the enzymes that you used to make your chimeric plasmid or your recombinant plasmid, you should get the sizes of bands that correspond with your original plasmid and the piece that you put in. So if you cut your plasmid with your enzymes, if you had your ampicillin resistance plasmid, you would get that 784 base pair piece because that's what's going to be popped out when you cut it with the two enzymes, and you're also going to get the band that corresponds to the remaining plasmid. Same thing with the kanamycin. If you cut with your two enzymes, you're going to get your one band, the 1,875 base pair band that contains the kanamycin resistance gene, and you should get the other band, which is the rest of that plasmid. So you should be able to add those up, and they should equal the total length of the original plasmid. So okay, you're going to take that chimeric plasmid now with ampicillin and kanamycin resistance genes, and you're going to transform it into E. coli bacterial cells. You're then going to take your E. coli that's been transformed, and you're going to streak it onto agar plate that contains both ampicillin and kanamycin, because you only want your plasmid that has both resistance genes to be able to grow up on that plate. So any E. coli colony that you see on that plate most likely contains your chimeric plasmid, or else the E. coli would not have been able to grow up in the presence of both ampicillin and kanamycin. So you can do the same thing with your clones that grow up on your antibiotic agar plates. So you can compare the banding sizes and the numbers of bands from your clones to that of your plasmids, and you can determine which one of your clones contains which plasmids and which pieces of DNA. You can also use phage vectors in addition to plasmid vectors, and there are many vectors you can use. Plasmids and phage are the more common vectors used. So using a phage vector, you would take your genomic DNA that you have cut with restriction enzymes. You would take your bacteriophage, which is a virus that infects bacteria. You can extract that DNA and cut that. You can ligate your cellular DNA with your bacteriophage DNA and make recombinant DNA. You can then place that recombinant DNA into a hybrid phage. You can take that phage and use it to infect bacteria. That phage is going to insert the recombinant DNA into the bacteria. The bacteria are then going to double. You can lyse the bacteria, which are going to form a plaque on your plate. So if you take your bacteria and you make a lawn on an agar plate, where you get bacterial lysis from your virus infection, you're going to get a plaque, and those phage clones are in the plaques. So you can end up with a library of phage clones. So here's an example of using recombinant DNA to produce insulin. So what you would do is you would isolate the gene for insulin from a human cell. You would then take that gene after you've cut it with restriction enzymes and insert it into an appropriate vector that have been cut with the same restriction enzymes. You're then going to take that recombinant plasmid vector, whatever vector you're using, and you're going to put it into cells that rapidly multiply, which is why E. coli bacterial cells are commonly used. So when you take that insulin gene and you put it into E. coli, that insulin gene is going to be expressed and actually make insulin. Insulin is going to be made and released, and you could then purify the insulin from the bacteria and use the purified insulin. So here is a schematic of how you would transfer and clone that insulin gene, and it's following the method that we've been discussing. Here's another example of how the Hepatitis B vaccine is made. So you would take a plasmid and you would insert the hepatitis B surface antigen gene into it and make your recombinant plasmid. You would then insert that into a cell that rapidly multiplies. In this instance, it's a yeast cell. You let that grow, you then lyse your yeast cells, and you then purify your hepatitis B surface antigen so that you can use that for vaccine. So recombinant DNA is what's used for gene therapy. Because in gene therapy, what you're trying to do is insert genes into individual cells in order to treat a disease. So for example, if you have a hereditary disease that has a mutant allele, your goal would be to replace the mutant allele with a functional allele. So gene therapy has been focused on diseases that are caused by single gene defects, for example, cystic fibrosis. You can use gene therapy to change the expression of a person's genes. You can target somatic or germ cells. And what you do is you insert your clone gene into the body through a vector. So gene therapy, commonly you're using virus vectors. So gene therapy using human cells and recombinant DNA is much more difficult than modifying bacteria. Usually because you have problems carrying very large sections of DNA and also making sure they're delivered to the appropriate site. So depending on what gene it is and what cells are abnormal, you need to get that gene into the appropriate cell inside the body in order for it to work. So that can be very, very tricky. So gene therapy research is aimed at correcting malfunctional genes to try to correct that malfunction. Vectors are used to deliver the therapeutic genetic material into the cells, and like I said, a common vector for gene therapy is a viral vector because viruses will infect cells and then will introduce their DNA into the cell. So if you have recombinant DNA in the virus that contains the correct gene, the hope is that the virus would introduce the DNA that contains the correct gene into the cell. There are problems with gene therapy though, problems with toxicity as well as immune and inflammatory responses. So for gene therapy, like I said, there are commonly viral vectors are used, such as retroviruses. Now when you're using a retrovirus, you need to make sure any dangerous portions of that virus that might cause a viral disease are removed. So you only want to use the portions of the virus that are going to be able to infect cells and deliver the genes that you need it to deliver. Adenoviruses, which cause the common cold, are also used. Adenoviruses will stimulate the immune system, but they do have a short survival. Non-viral vectors are used. However, non-viral vectors thus far have had very poor transfer. There's also, like we said, targeting issues where you have to get your gene into the correct cell, and if it's not inserted into the correct cell, it's not going to work, as well as controlling the gene. Using gene therapy, you really have to understand gene function. You have to understand how that gene works in order to make the appropriate recombinant DNA and try to get it into the correct cells. And the problem with this is that many genes have more than one function. So even if you know how that gene works for one aspect, you don't really know if it's going to be able to be used to treat that one thing, because it then might have a role in something else that may lead to a disease. So this can be a problem. Also, gene delivery. How do you deliver that gene, and ensuring that the gene is actually going to be expressed. The other problem is that gene therapy is really only good for monogenic diseases. And there aren't that many genetic diseases that are due to one gene. There's just a handful. Most disorders are multigenic. So there's many different genes involved, there's environmental factors involved, things like that. So gene therapy can't really be used to try to treat or cure every single disorder. Currently, there are well over 600 clinical gene therapy trials. Many of these are for cancer treatment, and although there have been some breakthroughs with gene therapy, thus far there have been no cures identified yet. And although recombinant DNA technology has done great things in producing chemotherapeutic agents, producing hormones to really high levels so that you could just purify them and get them in a nice form to use for treatment. There are concerns with recombinant DNA technology. For example, engineering food crops to resist pesticides. Some people are concerned about eating food that contains recombinant DNA because, what if the recombinant DNA gets into the body and causes a certain mutation and, things like that. Engineering microbes to produce deadly toxins. Now, in the research lab, and we engineer microbes all the time. We engineer microbes to be resistant to numerous antibiotics. So that's a concern because if those microbes that now produce a deadly toxin or are highly antibiotic resistant were to get out into the population, you now have this, super bug that can cause serious problems. There's also several other ethical issues, such as human cloning. We had the problem with, Dolly, the sheep cloning, as well as reproductive technology. So using recombinant DNA to change the outcome of, your offspring. So there are many, many ethical concerns with recombinant DNA.